ladies, today I'm going to go through the end of chapter 12 on linked genes. So what is a linked gene? Well, a linked gene are genes that are located on the same chromosome that tend to be inherited together. So because they're on different chromosomes, because they're on the same chromosome, okay, and not different ones, they don't follow the law of independent assortment that we normally see of genes that are on separate chromosomes, okay, because they're inherited usually together. What will happen is that when genes tend to be inherited together, you're going to get this variety of genetic recombination. So the production of offspring with combinations of traits that are different from either parent. So we have parental types, those ones that have the same phenotypes of the parent, but we're also going to have those recombinants, those recombinant types that are going to be slightly different from the parents. They're going to have the same traits as the parents, but they're going to be different combinations of those traits. And this is all the result of crossing over during prophase 1 of meiosis. Now let me show you how this works. Okay, so let's think about this for a second. So, oops, I need that. So if I was thinking about a traditional problem that I was going to solve, so let's say I am crossing together I'm going to cross together two homozygous parents. So say, what genes am I going to be using later? B and VG. Okay. So say I'm crossing, let me get my pen. Okay, so B plus, B plus, VG plus, VG plus. And the plus represent wild type, and that's the dominant trait here. And I'm going to cross that with a recessive individual, VG, VG. And all of my offspring that I'm going to get from this cross are going to be heterozygous individuals. Okay. If I then cross this heterozygous individual back with another recessive individual, okay, B plus, B plus, oops, undo. Okay, and note VG is one gene. It's for vestigial wings, actually, in flies. If I did this cross, what you would expect to see is a variety of different offspring. So remember, you do your foiling. Okay, so I have 4 different possible gametes that I could make. Make that Punnett square. So I could make B plus VG plus B plus VG, B VG plus, and B VG. Okay, and this individual right here is only going to be able to produce B VG. Okay. Okay, and note that this is just 2 genes. VG is for vestigial and just 1 gene. So it's going to be B VG. And when you go and you do this, there's going to be 4 possibilities. So B plus B, BG plus BG. Probably should have saved myself a little bit more room. BG, BG, BB, BG plus BG, BB, BG, BG. Okay, and when I'm looking here, I have, okay, this individual right here is going to be parental because it looks just like parent. Okay, it has a gray body and it has normal wings, I think. Yeah, gray body, normal wings. And this individual has a gray body a gray body and vestigial wings. This individual has a black body, normal wings, and this is black and vestigial. So I have 2. Okay, this one looks like his parent right here. Things are moving all over the place. Okay, this guy looks like this parent, this one looks like this parent, and I have 2 recombinants that look a little bit different. Okay, they're different combinations. If these traits were on different chromosomes, I would see a 1 to 1 to 1 ratio. Okay, just like how we've been doing normally in class. What's really interesting is that when scientists first discovered linked genes, they saw something a little bit different. Okay, so I'd have my gray body, normal wings parent, cross it with my homozygous recessive parent, black body, vestigial wings. I'm going to get that heterozygous individual. Okay, same thing I got here. So I got my heterozygous individual right here. Okay, if I take that heterozygous parent and cross it by the homozygous recessive, just as I did here, okay, I'm going to get that one gamete for my male who is homozygous recessive, and I'm going to get those 4 possibilities for my female who was a dihybrid. Note that because these 2 genes actually do occur on the same chromosome, you are going to see all 4 phenotypes just like you would see normally, but we're going to see it being expressed a little bit differently in the offspring. And how it's going to be expressed a little bit differently is that you're going to see a different ratio. You are not going to see that 1 to 1. Okay, 1 to 1 to 1 to 1 ratio anymore when you do this cross. You're going to see now a lot of parentals, and you're going to see very few recombinant offspring. Okay, and this is because crossing over, it has to do with how close the genes are on the chromosome and how likely they are to cross over and switch to produce these recombinants. Okay, crossing over doesn't happen every single time, but it does happen sometimes. And what we do when we look at our offspring from a cross, and we want to determine if it is possibly linked or if it's determine if we're seeing the ratio that we're seeing, we want to do a chi-square analysis and see if it actually is following this ratio or not. Obviously, with how distributed this is, it's not going to follow that chi-squared value, and the chi-square, and we would reject our null hypothesis because it is so different, because there really is a significant difference. But we also want to calculate a recombinant frequency to see where exactly these genes are and how likely it is that they will cross over. So our recombinant frequency is basically you take the number of recombinants over the total number of offspring. So in this case, we have 391 recombinants, and we have 2,300 total offspring. To get your percentage, you multiply by 100, and in this case, we get 17%. So what we do is you compare this to 50. So a 50% frequency of recombinants is observed for any 2 genes on different chromosomes. So if you had that 1 to 1 to 1 ratio that we typically see whenever genes are on different chromosomes, okay, just the normal problems we've been doing, if you see that ratio, you would have a 50% recombinant frequency. So if you think about it, Half of them would be parental, half of them would be recombinant, just like I said here. Okay, this one's parental, recombinant, recombinant. So you can see that because it's not following that pattern, we know that these genes are linked and they have to be on the same chromosome because they're not following the same pattern of inheritance. Okay, and we can determine how close they are by actually doing this recombinant frequency. Anything less than 50% would mean that the two genes are on the same chromosome. 17% is less than 50, so we know that they are on the same chromosome and they do not assort independently during meiosis. What we can do with that percentage is we can make a genetic map. So this is just an ordered list of the genetic loci along a particular chromosome. What scientists have determined is that if you have a higher percentage between two genes, that would indicate that they are farther apart on a chromosome, meaning that they're more likely to cross over because they're farther apart. If they're really close, it's less likely that you're going to have that synapsis taking place right in between them because it's a very small area. So what we can do is we take that percentage, that recombinant frequency, and we turn that into map units. 1% equals 1 map unit. And what you can do is you can put that on a chromosome. So what we were looking at before was the body color, and we were looking at the type of wings. So body color, type of wings. So this one would be vestigial. This would be a black body. You can see that they are 17% or 17 map units apart. If we did this experiment with another gene and looked at cinnabar eyes, which is what this stands for, you could see based on how body color and cinnabar compare to each other, this is 9%, and cinnabar and vestigial compare to each other 9.5%, we could organize these and try to figure out where they all align based on different inheritance patterns between different crosses. Last thing I want to talk about is just this new combination of alleles. So as always, when I relate everything back to evolution, this new recombinant chromosomes are going to bring a lot of variation, which is really, really important because variation allows organisms that have the best traits to survive and reproduce and pass on that trait to the next generation. So producing a lot of genetic variation is very favorable. Random fertilization of these gametes that do go through crossing over and have these crossing over events on the same chromosome is also going to increase the genetic variation of this population as well. Okay, you need to complete number 16 in your genetic practice problems packet. If you have any questions as you're going through that, please reference this PowerPoint or the end of chapter 12 and bring any additional questions or concerns with you to class. Have a nice day, girls.